good afternoon, everyone. It's glad to see so many people are still coming in. I'm Thomas Abil from TU Delft, and I'm happy to chair this session on Team of Genomes. We'll have three speakers, and the first one will be Shiraz Shah from Copenhagen University, talking about characterizing leader sequencing of CRISPR Leader.
Okay, yeah, I can hear myself now. Let me see here. All right, it works. And the pointer. There it is. All right. This is the first time that I've given a talk at such a big venue. By the way, how good of a place is this? I mean, what a wonderful venue to have a conference, actually. I think this is amazing. I feel almost as if this is my 15 minutes of fame in life, you know? That after this, it's all downhill, basically. Anyway, let's see what happens. So I'm going to talk about characterizing leader sequences of CRISPR loci. And basically, the first author of this paper is Omer. And this study is it's part of a long-term collaboration between two different labs. Omer, this guy here, he's from what I call the Freiburg lab, and they're a bioinformatics lab, and they work with RNA bioinformatics. And Omer's supervisor is this guy, it's Rolf Backhofer. And me, I'm right here, and I'm from the Copenhagen lab, and we work on archaeogenomics, and my supervisor is Roger Garrett. So we've been collaborating for a lot of years. And this paper here is just one paper in a string of papers, some of which are already published and some of which will be published, which focus on a lot of basic research regarding CRISPR systematics. So this is basic CRISPR research, it's not applied. So I think maybe some of you might get a bit disappointed because you're maybe here at the session due to the maybe potential applications, off-target prediction and stuff like that. That's not so much what this talk is about, though, so I have to disappoint you there, maybe. Anyway, let's get started. I'm going to give a brief overview of how the CRISPR system works. So basically, at the heart of the system, this is a prokaryotic cell, a bacterium or an archaea, as you can probably see. And at the heart of the system, what you have is the CRISPR-Cas locus, which is the genomic locus, and it's composed of a number of different distinct elements, all of which play a crucial role to the function. So you have the Cas genes, and the Cas genes can be divided into, the Cas genes can be divided into three different categories. There are the adaptation genes, the interference genes, and the processing genes, each of which encode their own protein complex, which carries out its own function. Then you have the leader sequence, and the leader sequence is what this talk is basically about. That's what I'm going to spend the next 15 minutes talking about. And then you have the CRISPR locus, or the CRISPR array. And what the CRISPR array basically is if you look at the DNA level, it's a series of repeats. The green ones are repeats. They're identical repeats, identical sequences, which go through all the way through the array. And in between the repeats, you have these white sequences. And these white sequences are not repeated. They're essentially kind of like random. They look random. People thought for a lot of years that they were random. They had no idea where they came from until three different research groups, independently of each other, found out that these actually correspond. They match exactly to viruses or phages which have infected the bacterium that carries this CRISPR array at some time throughout its history. So the way that the CRISPR system works is basically you have a phage infection. That's what initiates the whole process. You have a phage infection. The phage inserts its DNA into the cell, and then what happens is that the CRISPR machinery starts with the adaptation complex, which is encoded by the adaptation Cas genes. It recognizes the phage DNA. It cuts out a piece of the phage DNA, maybe 30 or 40 or maximum 50 base pairs, and it takes that piece and it inserts it in between two repeats inside the CRISPR locus. What happens after that is that this entire array is transcribed, and the CRISPR locus can be up to several hundred repeat spacer units. So it basically has a memory of the past several hundred infections, which is pretty impressive. Now this is transcribed, this whole piece, which can be several kilobases long, is transcribed into a long piece of RNA. And this RNA is then processed by the processing Cas proteins into small RNAs, each of which correspond to one complementary piece of RNA, which is complementary to the viruses that have infected throughout history. Now the processing complex loads this small RNA onto the interference complex. So this RNA gets incorporated into the interference complex, and the interference complex then starts scanning. It starts scanning all the DNA in the cell. If an infection by the same phage were ever to occur again, this interference complex will basically have detected that infection through complementary base pairing, and then it's basically going to cleave that target. So that phage is not going to be ever able to infect the cell again. So that's basically how the system works. There are several components, as I said, to the CRISPR-Cas machinery, and the CRISPR-Cas systems are found, as you know, both in archaea and in bacteria, which are amazingly diverse sets of organisms. And so the CRISPR-Cas machinery is equally diverse. You know, you have a lot of different types of CRISPR-Cas systems found in a lot of different types of bacteria and archaea. And they all have their specific components and their specific characteristics. Basically, if you look at the Cas genes, there's been made a classification of all CRISPR-Cas systems by looking at the Cas genes by the lab at NCBI, Eugene Kooning's lab. And this is the classification back from 2011. There's been an update of this classification in 2015. The reason why I'm showing the old one is because it's simpler. The new one is even more complicated. I mean, the old one we had three types, type 1, type 2, type 3. And now we basically have type 4, type 5, and type 6 added to that. And each type is divided into further a number of subtypes. So there's type 1A, type 1B, 1C, 2A, 2B, etc. All the way like that. So that's in terms of the Cas genes. People have also looked at the repeats, because the repeats also exist in different types and families depending on which organism you look at. Actually, Omer, the first author of this paper, has done a comprehensive classification of all of the repeats found in bacteria and archaea, CRISPR repeats, and he's divided them into superfamilies and families. Some of them are structured, and some of them are just sequence-based, which you can see here. The only component of the CRISPR machinery which has not been subject to bioinformatical scrutiny is the leader sequence. And that's because the leader sequence is really, really difficult to work with. I mean, it's not very conserved. So if you take a leader sequence from one bacterium and blast it, you're only going to find leader sequences from members of the same species which have also CRISPR locus. You're not going to go further into that. So that's why, and also it's really difficult to find the other end of the leader. Where does it, I mean, it starts at the CRISPR locus, but where does it actually end, and how much sequence are you supposed to include when you search for it? I mean, it's really difficult, and it's non-coding sequence. I mean, it's just DNA sequence, right? So it mutates really fast. What we know about the leader sequence is that it's non-coding. We also know that in length, it basically lies somewhere between 100 and 600 base pairs in length. It's required for adaptation. So the CRISPR locus cannot take up new spacers against viruses if the leader sequence is missing. This is something we showed in the Copenhagen lab back in 2006, and it's been shown in numerous other studies since then. It's composed of low complexity DNA sequence, so a lot of A's and C's and G's in a row, stuff like that. It's not conserved normally beyond the species boundary. At least that's what Mojica said in 2000 when he characterized the leader sequence for the first time. He said it wasn't conserved beyond the species boundary, and that's basically been, conventional wisdom in the whole CRISPR field ever since he said it, that you might as well give up looking for leader sequences because they're not conserved beyond the species boundary. BLAST and multiple sequence alignments don't really work because the sequences are too divergent. With MSAs, you get too many gaps. With BLAST, you don't get far enough. So those are the problems that we have to deal with when we look at leader sequences. One of the first, in-depth studies that were ever made on leader sequences was this study from 2009 from our lab. And what we did was basically we used repeat similarity as a proxy to gauge the similarity at the leader level. Because what we found out is that we had a series of CRISPRs in some hyperthermophilic archaea. We had maybe 10 or 12 different genomes, and they were loaded with CRISPR-Cas loci. And we took the repeats and we made a tree. And we could make a tree where we could define 3 major families of repeats. And when we look upstream of the CRISPR loci of each of those major families, what we found, we weren't able to align the leaders, but we were able to subject it to some kind of motif analysis. And what we found was that CRISPRs, or leaders pertaining to the first family, showed similar motifs, and leaders pertaining to the second family showed similar motifs, and so on. So obviously, these leaders were conserved even beyond the species boundary, contrary to what Mohica had said, if you use a sensitive enough method. And in this case, we used motif search instead of using BLAST or multiple sequence alignment. In 2011, we were actually able to make an alignment of the leaders, which was traditionally not possible, but this alignment is basically a manual alignment. So I basically sat here and I've inserted these gaps on the keyboard, like that, basically. And if you do that, you're actually successfully able to make an alignment. And by looking at this alignment, something becomes very clear. I mean, the leader starts up here, it's kind of going in reverse, right? Because it's on the wrong end of the CRISPR locus. So this is the first position right before the first repeat, which is somewhere around here. And so the alignment goes like this, and so we see that there's sequence conservation, weak sequence conservation all the way, and then all of a sudden the sequence gets random. So when it gets random, that's probably where the leader ends, right? And this is probably the boundary of the leader, at least that's what we predicted here. So we were able to define leaders in different members of Archaea. In a limited set of archaeal genomes, we were able to define all of the leaders and their boundaries using this method. And since then, there has basically been made no progress. This was back in 2011. So we showed for a number of Archaea that leaders did not only cross the species boundary, but they also were able to cross the genus boundary. I mean, this is Cephalobus, Acidianus, Metalospira. So across different genera of organisms, you can find the same leader, albeit in a very divergent, mutated form, which you can only align manually. So I was talking to Omer sometime in 2013. We were at a CRISPR conference, and Omer said to me, isn't there something that we can do with leaders? There must be something that you can do with leaders. And I said, it's too difficult. I mean, you have to do it manually. It's too much of a headache. And Omer, he insisted, he told me that we must be able to do it automatically. If you're able to do it manually, there must be an automatic way to do it. And once we do it, we're able to characterize all leader sequences in all bacteria and archaea. So we started working on that. And so basically, there are a number of, actually, due to time constraints, I think I'm going to skip this slide. I think the important concept here is basically that if you have a CRISPR locus, which end do you want to look for the leader? And of course you guys are going to say, of course you look at this end, right? You don't look at that end. But how do you know the orientation of the CRISPR locus? Because it's double-stranded DNA, how do you know if the CRISPR locus is on one strand or the other strand? Well, that's not an easy problem to solve, because these CRISPR repeats are palindromic. That's why it's called, that's what the P stands for there, right? So it's really difficult to determine what's the correct orientation. So this problem had to be solved first. I mean, this is an example of one of the repeats in Omer's database of CRISPR loci. And this is one family of repeats, and as you can see, it has a palindrome there, which forms a hairpin. But apart from this palindrome, I mean, even the ends are reverse complementary, CAAA, GTTT. So how do you know what is the correct end? And this is actually quite a complicated problem to solve, and Omer, he managed to solve it using machine learning, and he published that in a paper on which I was also a co-author 2 years ago, also at an ECCB conference where he presented this actually. So after solving the repeat orientation dilemma, it becomes much easier to actually solve the other dilemma, which is finding the leader. So basically after that, we have to define a cluster of repeats for which we know that the leader will be sufficiently similar for us to detect sequence similarity and find the other end. And this is the total diversity of CRISPR repeats. I mean, most of CRISPR repeats ever found have a very common motif in this end, GAT-GAG, which I used to call it, and the other end is not that well conserved. If you look at one of Omer's families, one of his 24 families of CRISPR repeats, this conservation, there's a little bit more conservation here, but that's still not enough to find leader sequences. We need to go all the way to what we call the cluster level. And the cluster level, a cluster basically comprises of a set of CRISPR repeats which are 95% identical to each other. So those will be CRISPR loci spread over different bacteria and archaea. As long as they're 95% identical to each other on the repeat level, we put them in the same cluster. And then we're actually ready now to detect the leader sequence. And this 95% cutoff is something that we've actually optimized, so to speak. So if you look at a region 600 base pairs upstream of a CRISPR locus after having determined the orientation, and after ensuring that all of those CRISPRs are within the same cluster, then we can start saying, okay, where is the leader here? It must start here, but where does it end? And since you can't use multiple sequence alignments, what Omer did instead was to use windows where he counted k-meres, and he normalized those k-meres against a randomized version of each window, and then you can basically make a graph which iterates over the length of the leader, and as soon as the conservation starts dipping, that's probably how long your leader is. But since individual leaders may have indels, insertions and deletions, you don't actually know whether each individual leader is that length. They're just approximately that length. So what we do is we extract the sequence corresponding to this area plus a little bit extra, and then we do a traditional multiple sequence alignment, and then we go in and look where does the conservation stop, and that's where we set the cutoff. So that's the length of all of those individual leaders minus the different gaps that have been occurring. And using this method, we're able to identify leaders in most bacteria and archaea. I'm going to go through the results now. I'm almost at the end of my talk. So from the 3,700 CRISPR loci in Omer's database, we were able to find 195 different leader clusters comprising 2,993 different leader families. So from the vast majority of CRISPR loci in Omer's database, we've been able to find corresponding leaders, and we've determined their length quite accurately. Here's a table showing basically, there have been some people who have determined the length of leaders in a number of different model organisms, including us down here. And this is what they report in their studies. This is what our method predicts, and this is the difference. So in most cases, it's less than 10 nucleotides. So the method works pretty well. One of the most intriguing results about this study is if we look at bacterial leaders, this doesn't for some reason apply for archaea leaders. But if you look at bacterial leaders, the clusters are vast. These leaders, this is the same leader found in a number of diverse different bacteria, many of them are from different phyla. The phylum is the phylogenetic hierarchy right below the domain, right? So basically, all the bacteria can share the same leader across each other, albeit in highly diverged form. But you can see how low the sequence conservation is in this particular family of leaders. But there are some striking motifs here and here. This is a leader. This is one cluster of a leader which is found in a lot of different bacteria. In fact, if you look at the top 10 biggest bacterial leader families, we see that most of them are shared across several different phyla. These are the different bacterial phyla here. If we look at the archaea, this is how the image looks. Archaeal leaders are not shared between different phyla. Either they're Trenarchaeal or Euryarchaeal. These two phyla are really small, so they didn't make it into the top 10, basically. So this is a very stark difference between archaea and bacteria. We don't really know why. So if we look at basically the implications of this result, we can see that Mojica predicted back in 2000 that leaders don't cross the species boundary. So that's where we're stuck. Now, in 2009, we showed that we were able to find leaders within the same family. And in this study, we've been able to find archaeal leaders which go all the way up to the order boundary, but bacterial leaders are all the way up to the domain boundary. They cross the phylum boundary, right? If we compare this to the diversity of, for example, type 2 systems, type 2 CRISPR-Cas systems also are able to cross the phylum boundary, but you don't have type 2 systems in archaea, so they don't cross the domain boundary. So they're kind of stuck here. And type 1 and 3 systems are all the way up, crossing also the domain boundary, because they're found both in archaea and bacteria. So that's basically it. So as for the conclusions, this is the first automated method for determining leaders. And we have a command line tool, and we have a website where the results are available. We're now a step closer to a fully automated method for annotating CRISPR-Cas systems on genomes of archaea and bacteria. And we find that leaders are wisely conserved across different bacterial taxa. And the fact that they're so conserved, it must mean that the underlying mechanism of adaptation must also be conserved. Because the leader was the joker in this whole thing, right? We know about the Cas genes already, the repeats and everything, but the leader was kind of the variable. Now we know the leader is also conserved, so the adaptation mechanism is probably conserved. So that's basically it. I'd like to thank everybody who participated in this study, especially Omer for letting me give this talk. I'm so grateful for him letting me do this. And I'm also grateful for the organizers. giving me this opportunity, and I'm also thankful for members of the German 4-1680 Consortium who have funded some of this research. Thank you very much.
Thank you so much. I think we have time for one burning question, if there is any such question. No burning questions?
You can also ask a question generally about CRISPR, if this is too nerdy.
So I do have a simple question. So you said that the interference complex is scanning all the DNA. How does it avoid essentially eating itself?
Yeah, I mean the spacers in the CRISPR locus have to be sufficiently different from any host locus. And if that's not the case, then it will eat its own DNA and the cell will die. So that does happen. It happens very often, but those cells get selected against, so they never live to see the day of light.
Okay. Let's thank Shiraz again. So the next speaker is Yanni Sun from Michigan State University. She'll be talking about assembling